Mitosis is the type of cell division that results in two daughter cells, each having the same number and same kind of chromosomes as a parent nucleus, typically of ordinary tissue growth.
Interphase is the prephase of mitosis. It is the longest stage of the cell cycle, and the cell spends more than 90% or more of their lifespan in interphase. During interphase, the cell takes in nutrients, manufactures proteins and other substances, expands in size, and performs a special function. Interphase is a preparation for cell division. This is when it copies all of the DNA molecules, and the nucleus consists of two connected copies called sister chromatids. In animal cells, it also copies the centrosomes. It starts with the G1 phase, which is the first gap phase. This is when the cell grows larger physically, copies organelles, and makes the molecular building blocks that are needed in later steps of mitosis. Next is the S phase. The cell synthesizes a complete copy of the DNA in its nucleus and duplicates a microtubule organizing structure called the centrosome. Lastly, the G2 second gap phase occurs. This is when the cell grows more, makes proteins and organelles, and begins to reorganize its contents. In the G2 phase, once it ends, is when mitosis begins.
Now we'll begin with prophase, the first stage of cell division. In this stage, duplicated material carried in the nucleus gets separated, chromosomes become visible, and the nuclear envelope disappears at the end of this phase. The DNA proteins known as chromatin condense, and this starts the beginning of the substage prometaphase.
Metaphase is the third phase of mitosis. The spindles have captured all chromosomes. All chromosomes are lined up down the middle. Two kinetochores from each chromosome should now be attached to microtubules from opposite spindle poles. Before the process concludes, the spindle checkpoint will occur, ensuring all chromosomes are at the metaphase plate, with their kinetochores correctly attached to the microtubules. To ensure the proper cell division, if there are any problems, the cell will stop mitosis until it is fixed.
Anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Before anaphase can begin, sister chromatids are aligned in the middle of the cell. Once the paired sister chromatids separate from one another, each is considered a full chromosome. They are then referred to as daughter chromosomes. Through the spindle apparatus, the daughter chromosomes move to the poles at opposite ends of the cell. The spindle apparatus is made of many long proteins called microtubules, which are attached to a chromosome at one end and to the pole of a cell at the other end. In preparation for telophase, the two cell poles also move further apart during the course of anaphase. At the end of anaphase, each pole contains a complete compilation of chromosomes.
Telophase occurs as chromosomes reach the poles and the cell is at the end of its division. The spindles and asciters of early mitosis disappear, and each are forming new cells that begin to synthesize its own structure. Nuclear membranes begin to enclose the separated chromosomes. The coiled chromosomes unwind, becoming chromata once again. Furthermore, the cytoplasm breaks into two separate bodies, but it still continues to be only one cell.
Mitosis is followed by cytokinesis. Cytokinesis, also known as cytoplasmic division, is the process in which the original cytoplasm is divided into two new daughter cells. This begins prior to the end of the anaphase and is completed shortly after the telophase. The chromosomes normally are equally distributed between the daughter cells, both containing 23 pairs of chromosomes, having a total of 46 chromosomes altogether.